Non, mais je d'accord. And this information can be used for conservation. And we work. I won't talk about this today, but we work, for example, with the Castelin National Park about the evolution of this species. So today I will mainly talk about 3 topics, more or less developed. The use of genetic markers for barcoding of species to study species limits, the question of genetic structure, and a bit of adaptation. Very low adaptation, in fact. So if we talk about genetic markers, I don't know if some of you are very familiar with that, maybe not all of you. So crude separation could be the case of the use of low number markers. That means we analyze one or few regions of the genome. So we had the single locus sequencing, for example, mitochondrial DNA, which has been used and is still used for barcoding, for example. We had microsatellites. We'll not talk about that today, but they can still be useful. We'll see a bit of them. And we are now in the era where we more and more develop high throughput approaches, which leads to the study of, for example, SNPs, which are variation of one nucleotide in the genome. So either you do whole genome sequencing and you have some idea of the variation at the genome scale, or you say, for example, okay, I don't have money, time, or I don't have interest in analyzing the whole genome, and you say, I will analyze only a part, for example, 5% of the genome. And if you do that, you want to have some techniques which allow you to have analyze the same 5% in your different samples. Because if you analyze 5% on one side and different 5% in another part, in another sample, you can't compare them. So there are different techniques which can be used to do partial genomics, I would say, or reduced genomics, which are rad sequencing, target sequencing, transcriptome sequencing. And we will see some of these approaches too. Okay. So the first topic where I will present some of these techniques is about barcoding and species limit, which are different questions. Here is our unicellular gorgonians from the Mediterranean. So in Anthozoans, not only, but there are potential difficulties for taxonomists, where in some cases we have few morphological characters. We have seen some of them today or these days. We have some cases of morphological plasticity. In corals, we have several cases for that, and also a case where we have hybridization, which can blur the differences between species. So the molecular markers are very interesting in integrative approach, which means taking the information not only from the genetics, but also from the morphology or other characters. So what molecular markers can we use in Anthozoan? The first studies, here I cite Shearer and collaborators, but others in the 2000 when we were doing the sequencing of mitochondrial DNA, like other people, we have seen that there is a slow mitochondrial evolution in the Anthozoan. That means that in some cases, which is not always the case, I will show you that we can still use mitochondrial DNA, but there are no or low variability of mitochondrial inside the species. That means that in some cases, mitochondrial DNA was identical among the different samples you see for one species, and which is more problematic if you want to use it for barcoding, for example. We have very low divergence, very low differences between species when compared to their metazoans. So just to mention, it's not specific to Anthozoans. For example, this is also observed in sponges, for example. So what markers can we use for barcoding of species delimitation? So just as an illustration of this, some species on which I work since several years, if we look at the traditional barcoding locus, which is mitochondrial CO1, and we don't have any differences between these species of the same genus, which is here Unicella singularis and Unicella cavogni, there is no Veracosa anyway, which are morphologically well separated, but there is strictly no difference for the 600 base pair of CO1 we usually analyze. So what can we do? There are several answers to that. One of them has been proposed by Catherine McFadden in this very important article, if you want to work on that, which has been proposed to use extended barcode. That means not only analyze CO1, but also analyze an intergenic region, which is close to CO1. So it's all mitochondrial DNA. And another locus, which is in mitochondrial DNA and which is specific in mitochondrial DNA of octocorals. Only in the mitochondrial DNA of octocorals, no other animals have this locus, which is mitochondrial mutes or MSH1, depending on the paper who use it. So this extended barcode is well used in octocorals right now, and you have a lot of sequences of CO1 IGR, mitochondrial mutes, or sometimes both of them for the same species. So I will go fast about this, but it was in this article of Catherine McFadden, which has shown the mean and of the genetic distance of percentages observed within species and within genus for CO1 alone, CO1 with IGR and CO1 with MSH. I won't stay there, but just this is another representation of that with histograms, which present either CO1 alone, MSH alone, CO1, IGR, MSH, the extended barcode, which present the distribution of the distances inside species or between species. And just here it's the zero. That means you have no difference. And the idea is that if you combine these different sequences, you still have some cases where you can't separate the species. For example, for our Unicella, it will stay the same, but it's reduced. So it means you have a more you are more powerful to discriminate some species with mitochondrial DNA if you use these three mitochondrial regions. One thing to keep in mind is that these are all mitochondrial, so this is not independent information, right? Just to mention also briefly that if you use this, you can do some modification of protocols. It's the only place where I will talk about protocol variation, but just to mention that the initial article is also very interesting to look at precisely because you have different primers which are proposed to analyze this locus. That means if one primer pair, which you use in PCR, doesn't work, you can try other ones. The other thing is here you have an example of a plate of DNA extraction after digestion of great diversity of octocorals from the Lenguru expedition in Papua. And just to see that you have different colors which correspond to different sclerites, different secondary metabolites. And when we did that, we also had to, in some cases, the first primer did work, but in some cases it didn't work. So we had to change primers. And other things which can be useful in some difficult cases is to try to dilute DNA, to use BSA in your PCR. And this can be interesting depending on what you do. So here it's a case where we had very wide diversity of octocorals. But just to mention that, yeah, it can make successful something which it didn't work. What about hexacorals? Generally, we have low variation of CO1 as well, but in some cases it works. So mitochondrial DNA can still be useful with CO1, with another locus I did not mention here is 16S, for example. But there are some specific cases where mitochondrial DNA is informative. One of these cases is the case of Pocillopora corals, where since several years, people use mitochondrial ORF locus to do some barcoding and some species delimitation. So ORF means open reading frame, so it's really not specific for locus. So in fact, this corresponds to this locus apparently, TMP 362 gene in the mitochondrial DNA. I don't remember what is TMP 362, but if you want to have a look. Why Pocillopora? Because in Pocillopora there was a great mess in taxonomy. The number of species which was described varied a lot among the authors, and typically in Pocillopora you have all the problems I mentioned before. You have morphological plasticity. The same colony in different environments can have different morphology. You have a case of hybridization and so on. So here it's I mentioned just to give an insight of what we can do. It's an article from the PhD work of Pauline Gérin, who worked with Hélène Magalon at the La Réunion University. And she made a great job at that time sequencing mitochondrial ORF. And you don't see very well, but I just present part of the tree which can be obtained. With this tree, she tried to apply different methods of species delimitation. I won't talk about that today, but here you have a team who is very expert in species delimitation. And now, based on this, they proposed several species hypotheses, which are not primary species hypotheses here, which correspond to group of sequences well separated one from other. So here they propose some species delimitation on the basis of the variation of mitochondrial ORF. And this gives also a database when you have new samples to compare these samples to this one for further identification of new material. Just to mention that in this first article, they also tested the validity of this hypothesis with other markers independent from mitochondrial DNA, which are microsatellites at that time. So that's already said. So these primary species hypothesis will be tested with and refined with nuclear markers, and I will talk about that just after. I will go fast on that, but just to mention, if I go back to octocorals and the low evolution of mitochondrial DNA in octocoral, that it's not always the case. And because since it's a quite recent article from last year, where some authors have observed two things. First, there are one or two cases where the mitochondrial DNA doesn't have mitochondrial mutase. I didn't mention that, but mitochondrial mutase is a gene which is involved in bacteria in DNA repair. So there was a nice story to say, okay, there is mitochondrial mutase in the mitochondrial DNA. It makes DNA repair, and if it repairs DNA, there is a low mutation or a low number of mutation, and that could explain why mitochondrial DNA doesn't vary in this species. The story is very nice. Here we see that if you don't have mitochondrial mutase, you have this very long branch for this octocoral, Pseudo-Autobastus, which could say, okay, we don't have mitochondrial DNA, and this long branch means a lot of mutation, so it would say there is a lot of variation. But you have other examples of long branches, which means a lot of mutation, accelerated mitochondrial evolution, for species which still have mitochondrial mutase. So the story is nice, but probably doesn't explain everything. And remember also that you have other species. I talked about sponges, some hexacorals, for example. You have other species which don't have mitochondrial mutes and which also have a low evolution of mitochondrial DNA. So I would say that this is part of the story, but this is not the whole story. If we want to go a step further, I started to talk about that with Pocillopora. We have to find other loci because, okay, we have seen that mitochondrial DNA is often not very variable. Usually it has a peculiar transmission, uniparental, but for some species, strange species, Eric like that. And the fact also is that if you want to, not only to do barcoding, but if you want to delimitate species, see what are the differences among species, it's important to use multiple independent genetic markers, independent loci. So in the literature, we have seen that there is a range of markers which have been used, and I will present some things about nuclear SNPs today. Cette page contient une erreur. Okay. So I will show you what sequencing. Okay, so there is a figure here which presents what sequencing. You see very well. So just to explain what is rad sequencing is the idea that you will not sequence the whole genome as I explained at the beginning, but you want to sequence part of the genome. So you will cut the genome with restriction enzyme. This enzyme will cut the genome at specific sequences. So stretch of sequences around 6 base pair, for example. And you will sequence the DNA on both sides of these short sequences. So if you do that, you expect to sequence, for example, as I said, 5% of the genome, specifically where you have these sites, where the restriction enzyme will cut the DNA. With that, you can compare these short sequences among the different samples you analyze, either samples from the same species or different species. And it's very useful because you can get hundreds to thousands or tens of thousands of independent markers in non-model species. When I say non-model, that means that you can do this in species where you don't know the genome, but it's better if you have a genome, the analysis is a bit easier. And it can be used in different topics. We'll see some of them today: population genetics to species delimitation. And in some cases, phylogeny. There are a few papers on phylogeny, but we can discuss about that, or you will talk with Eric. If it's very divergent, usually it's no more informative. So just an example of the use of rad sequencing to the question of species delimitation. So it's the article of Eric about Chrysogorgia octocorals, where, so you correct me if I will go fast on that, but where he compared the information gained with mitochondrial DNA, here with the mutes locus I mentioned before. And with the analysis of 91 colonies, he got 12 different sequences, 12 haplotypes. And with rad sequencing, he got, depending on the samples and the way you analyze the data, from a few markers to more than 2,000 markers. And is it okay? In most cases, you had a congruence between haplotypes and the rad clades in saying this is identical or this is different. These are potential same species or different species. But with a few exceptions, with one case where mitochondrial haplotype said it's the same thing and rad sequencing said it's different things. And on the contrary, one case where we had Similar things with rat DNA, rat sequencing, and mitochondrial haplotypes were different. And this is very interesting because we go back to what I said that, in fact, we had the idea of what we usually say, including myself, that mitochondrial DNA has a low evolutionary or slow evolution in octocorals, but there are some variations. Another approach is what we call target enrichment and sequencing. I will talk about that, about octocorals. So Eric talked about that this week, about these ultra-conserved elements. So the idea here is that you will sequence DNA, you take the whole DNA of a sample, but before sequencing, you will enrich DNA in what you want to analyze. So for that, you have first to define some targets. What do you want to sequence? So here, these are ultra-conserved elements. That means part of the genome, which do not vary much among species at various levels of phylogenetic diversity. And also here in the original article, there were exons, so the coding part of the genes. And from these targets, you will define what is called baits, which are short stretch of DNA that you will use to, what I would say, fish or catch these UCE and these exons in your DNA. So you catch, you enrich your DNA solution with these loci, and then you can sequence them. There's various interest in this. First, it's very robust, more robust than rat sequencing, and you can work, for example, with a wide range of species of taxa, if you can define this, and you can also work on degraded DNA. So this is interesting, for example, for some old samples in collection and so on. And this can be used from species delimitations to phylogeny. It's more interesting than, in fact, rat sequencing is not useful for deep phylogenies. So just to remind you that all the discussions we have about the new definitions of families, plus by the last article of Catherine McFadden, is based on these phylogenies, which are built with these kind of markers. So this is just a small figure about what we do. If you have your DNA, which is fragmented before analysis, you have some short strands of DNA, which will be used to catch the DNA you want to sequence. And this can be attached to magnetic beds. And then with the magnetic beds, you keep mainly the DNA of interest, and you can wash the other DNA, and then you can work and sequence mainly the DNA of interest, mainly. So just to a bit of numbers. In the initial articles, the initial bait set prepared by Andrea Quattini and Catherine McFadden, at the scale of Anthozoans, there were almost 1,800 loci shared among UCE and exons. And now, they change a bit these ones, but you have specific hexacorals and octocorals bait sets. That means that if you want to work specifically on hexacoral, you have one bait set, or octocoral another one. And they are all available at one company, which is Arbor Bioscience. I don't have any financial interest in Arbor Bioscience, but just to mention that. And the interest also is that there is possibility to subcontract the analysis, preparation of libraries, and sequencing. An example of application is work made during its PhD by Nicolas Houry. He worked with Hélène Magalon again. He has done a very, very nice PhD, by the way. And again, in Pocillopora, he started with a huge sampling set where they have more than 9,000 Poecilopora colonies from the Indian and Pacific Oceans. They made some sequencing of mitochondrial ORF, the gene I mentioned before, on 10% of the colonies, which gave a primary species hypothesis, a primary species delimitation. And the idea was to test this with the target sequencing I mentioned before. So they did this on a subset of this because it takes some time and takes some money. So more than 300 colonies chosen among the most divergent ones. And he analyzed more than 2,000 loci to test the hypothesis. Again, I won't go into details, but here you have this tree, the phylogenetic tree of the different samples. So with the tree, you can already have some test of species limits, what can be the same species or different species. There are additional tests which are, for those who are familiar, but I will present other results later, some analysis of genetic structure on this same data set to see, okay, is it homogeneous or is it a bit, for example, here we have an individual which maybe is mixed with others. And here you have all groups. So this is part of the tree, which is much bigger than that, but okay. If we compare with morphology, mitochondrial DNA, what can be the main information? So it's part of the results here. So I did not put the whole table, but at the end, they got, among all the Poecilopora sample analyzed, 13, I would say, robust species hypotheses. And one main result is that in Poecilopora, but I think it was well known by different researchers, macromorphology can be misleading. But if we go into micromorphology to analyze the polyps, the skeleton of one polyps, and so on, there are some characters which are interesting. And this is very important because in his paper, Nicolas, okay, he did great molecular stuff, genomic analysis, but he also went to see the micromorphology of the colonies. And there are some characters which are interesting to see, to look at when you analyze your samples. If we go back to mitochondrial DNA, okay, now he has a lot of markers, has a big stuff of almost genomic sequences, but mitochondrial ORF, finally, is still diagnostic for most species. So it can be used as the first step if you want to analyze your data to have a first idea about what kind of Poecilopora you have in your samples. Another stuff which is very interesting now is that still, as micromorphology is not informative, it means that what we have about the distribution, the range of a lot of Poecilopora species may be wrong, because we don't really know what has been analyzed. So now we can have more information or more precise information on the distribution of species in this genome software. What's next? Next, it's to analyze whole genomes. I will not talk about these articles because I didn't have time to really talk about it, but to really read it and so on. But it's a part of Tara Corail, I don't remember, one Tara expedition dedicated to coral reefs by Chris Woolstra and also Didier Forcioli at Nice, where they used some sequences to analyze the differences among Poecilopora corals, but not only at the whole genome. In fact, it's not about genome. They took some samples of corals and they sequenced everything. When I say everything, it's the microbiome and also the genome and everything. And so in this bunch of sequences, they took sequences of corals, and with this information, you can analyze the differences among the Poecilopora samples almost at the whole genome level. What is the interest? Because you could, if I go back to the Poecilopora, you've seen that, okay, we had mitochondrial DNA. With microsatellites, it was 13 or 15 microsatellites, maybe. You can still test this first hypothesis. With target sequencing, we see that, okay, we can separate. So is it useful to have the whole genome? In fact, the whole genome is useful to go further on that, but also to study, for example, the adaptation of these species to different environments. Another thing which is useful when you use a very much higher number of loci is to study what I would call speciation processes. And I will talk today briefly about that with the case of Dendrophilia corals. So if I talk about speciation processes or speciation histories, if you have two samples or two groups of samples that you see that you have differences among these samples, and these differences, maybe you are able to say that these are different species. Okay, maybe these are different species, but maybe the history among between these different species can be quite variable. So, for example, of different histories of divergence or speciation, which can be observed. The simplest is strict isolation. It means that you have an ancestor, then, for example, they are separated, and you have divergence with genetic drift, mutation, maybe selection. No more gene flow. Okay. Another history could be ancestral migration, which means that, okay, there is a first divergence, but there is still a bit of exchanges, a bit of hybridization, gene flow, and then no more. Which means that now, currently, you don't have any more gene flow. Another model which has been studied for quite a long time in some papers is isolation migration, which is more complicated to study. It's the idea that you could have divergence, but still a bit of continuous exchanges. In some cases, you have hybrids. And then another one is secondary contact, which in fact has been observed in various cases, where you have divergence first, for example, in different areas, but at some time, the two populations, maybe not already species, can be present in the same area and can form hybrids. Okay, so that means that in this case, you had a separation, accumulation of divergence, but apparently there is still the possibility to produce some hybrids and to have some genes going from one direction to in another direction. So it's very interesting to study that, to better understand how the speciation process takes place, and to study the evolution of the differences at the genome level. I started to do that with the case of Dendrophilia corals in the Mediterranean. So here you have just a distribution of the two species we were studying. In red, Dendrophilia cornigera, and in blue, Dendrophilia ramea. So these are not the sampling sites. We use much less samples. And we were specifically interested in some corals discovered in 2016 or 17, I don't remember, in Cyprus at 130, 150 meters, for which at the beginning there was some doubt about whether it was Dendrophilia ramea or cornigera, because the morphology was a bit weird. It seemed to be Dendrophilia ramea, but it appeared quite elongated, a bit slender, so there was some doubt. So what did we do? First, as usual, a bit of mitochondrial DNA. With CO1, yeah, we see some differences between cornigera and ramea. Not a lot, I think, if I remember well, it's 2 mutations, maybe, but okay. these are fixed differences, and the mitochondrial DNA indicates that, okay, Cyprus is really Dendrophylla ramea. Then I sequenced the transcriptome, which means all the expressed genes, the genes expressed by these species. So it's just a part of the genome, it's what is transcribed. In fact, you get messenger RNA and you sequence all of this. And if you get all these sequences, two informations. First, there are very clear differences between Cornigera and Ramea. And second, okay, the corals from Cyprus are indeed Dendrophylla ramea. So this is okay to try to say what is it. But the idea is also to test the scenario I mentioned before. What is the history between Dendrophylla cornigera and Ramea? Briefly said, what do? You, of course, use a sequence. So it's transcriptome, but it can be done with other data. What you need to have is enough independent genetic markers. So usually nuclear markers. I mean, this, you can do it, but it's useless. It won't be informative if you use one locus, one mitochondrial locus. And I'm not sure even if the pipeline will work, but it's not informative. You need a lot of loci. You have to clean the data to be sure that the variation you observe is robust, that there are no sequencing errors and so on. And you compute, there are some pipelines to compute some statistics about the diversity inside each sample, the differences among samples, and so on. But say it seems quite like a black box, but you have, I don't know, maybe 15 different statistics which can be computed. Second, in fact, I use this pipeline, which is called DILS. DILS is for demographic inferences with linked selection. And this pipeline will do simulations under different scenarios. The 4 scenarios are presented, but not only. And for each scenario, it will simulate the kind of genetic data you have, and it will compute the same statistic. And then, at the end, we will try to see if one scenario is better or is the best to give the same, for example, the same diversity or the same differences between samples than your observed data. And starting from that, you can try to see if one scenario is really better than the other ones. So in the case of Dendrophylla, the observed scenario was the simplest. It was strict isolation. That means that we have the separation. We don't have current gene flow, so no more hybridization. And the interest is that you can also have some estimates of some parameters. For example, we can try to estimate the date at which we had a divergence among our samples, and in this case, it was 4 or 5 million years ago. Okay, so last part, I realized I was a bit fast, maybe, so do not hesitate to stop me for some part. Last thing is about population genetics, which is what I've done since a very long time. And I will talk about this species, which is one of my favorite species, which is the Mediterranean red coral, where you can see, we'll see a better picture later, but here you have the skeletal axis red, and here you have well-expanded polyps. You have an activity of opening and closing of polyps in this species, so you have sometimes where it's open, others where it's closed. So what are the objectives of population genetics? Again, it's very crude, it's very synthetic, but for example, you can use this to estimate genetic diversity, which is important because that's what I always say to my students and what we could say to some politics in some case. But when you talk about biodiversity, to preserve biodiversity, in biodiversity, there is also genetic diversity. we'll see that it can be used to study the reproduction, to analyze the genetic structure, to make some inferences about the size of population and the level of exchanges among population, and also to study adaptation. So regarding reproduction, these are just examples of what we can do, but you'll see that there are some implications for collections as well. I go back to Pocillopora corals. So again, a shallow species. But in Pocillopora and in other corals, depending on species and sometimes maybe on population, the reproduction, okay, in all cases they can do sexual reproduction, but there are some cases of asexual reproduction. And among asexual reproductions in Pocillopora, there are various articles about clonal reproduction. That means that in some cases, if you have a colony of Pocillopora which is broken, the two parts can still live, grow, and they, as they come from the same colony, they come from the same zygote at the beginning, so they are almost genetically identical, so these are clones. And so you have clones. You can have clones in the same population, sometimes at variable distance. And these levels of clonality can be very different depending on the environments. So there are some studies depending on, for example, the strength of the waves and so on. To study clonal reproduction, we could say that it's easy. If you take two colonies, you can try if they are genetically identical. But if you do that, you don't analyze one locus, because by chance the two colonies can be identical with one locus. But if they start to be identical at 2, 3, 10, 20, 100 loci, 100 genetic markers, that's very strong information indicating that these are clones. Okay, so the more genetic markers you analyze, the more powerful you are to be able to say that yes, these are clones, or no, these are probably different colonies from different sexual reproduction. So for that, microsatellites are very useful, and with, depending on their variability, with 10 to 20 microsatellites, it can be powerful enough to separate the clones. But it doesn't give you information about the consequences of clonality on the whole genome. So we did some analysis of the impact of clonality in the case of Pocillopora in a study where we used rad sequencing, and we analyzed two sites. One in the Gulf of Oman, and the other one in French Polynesia. Strange sampling, by the way. In fact, these samples at the beginning, at the very first, they were not used to do population genetics. They were dedicated to the study of the adaptation to different thermal environments. Okay, but we had them, so we said, okay, can we analyze at the genomic level the differences among the two areas and among sampling sites? Just to say also that first we sequenced the mitochondrial ORF to check if there were the same species. So we had 3 main mitochondrial lineages. In Oman, in Oman 3E, 3G, 7A. So these are different sequences of mitochondrial DNA, but according to Pauline Gélin and so on, these could be the same species. And in Polynesia, we had another lineage, 5A, which is probably a different species. When we use rad sequencing, we will not present this, but okay, this was very clear, the distinction Oman, French Polynesia. But in Oman, we had these three ones, so we could test with mitochondrial, with rad sequencing if these three ones could correspond to different species. Okay? The corals with this mitochondrial DNA, do they correspond to something different from this one and this one? So with rad sequencing, I got something like that. So maybe you're not familiar with this. It's we really love that in population genetics in several years. But, okay, each bar is a sample, a colony of Oceopora, and the color corresponds to a genetic group. If a colony has almost all green, that means she belongs to this group. And some colonies can be, for example, this one could be shared, could be mixed, that's the better word, between two groups. So maybe it's a reproduction between the, what's this color, purple, blue, and green genetic group. Okay. So what did we get? So all these samples are from Oman, not so distant sampling sites. This is almost all green, so it seems there is a dominant genetic group. And there are some colonies which are a bit different in this. Strange. First test, do they correspond to different mitochondrial lineages? I guess that it's not easy to see, but here we have mitochondrial lineages. So if you are very powerful, I'm not, but trust me, the mitochondrial DNA does not explain these differences, okay? So if I go back to the mitochondrial DNA, for me, there are no differences among the main mitochondrial lineages in Oman. So how can we explain these different samples? We analyzed a bit more the differences among samples. So I try to explain that. Here is the distribution of the percentage of differences among samples. I analyzed, don't remember, well, let's say 10,000 leucine, 10,000 genetic markers. And I try to count the percentage of differences among the samples. So I take all the samples 2 by 2, we compare them. And here we have a bunch of differences between most samples are different by 10 and 15%. Okay. But we have also some samples, not so numerous, where you have very low differences, very low level of genetic differences. What is this? But in fact, these are clonal lineages. These are clones which are present in our data. You may say, okay, if these are clones, we would expect a divergence of 0. If there are clones, they should be genetically identical. If you use rad sequencing, you don't get 0. Generally, you will never have 0 divergence. For 2 reasons. First, when you do some rad sequencing, there are some sequencing errors. So even if you get the same sample, you sequence it twice, you will get some differences. The other reason is that if the clones are old enough, you could have some mutations, a bit of mutations, which have present, which can separate the different colonies which come from the same clone. So this is exactly what we expect from clonal lineages analyzed with rad sequencing. And another way to present this is a kind of tree. At the individual level, it's not a phylogenetic tree. Each branch is a sample. So most samples are well separated, but in some cases, it's not easy to see. These samples, for example, they are connected with very short branches, which means that they are very similar. And we confirmed that these are indeed clones because we had other information. We use rad sequencing, but also for some samples, we had microsatellite data. And for microsatellite data, indeed, as we analyze only 10 or 20 loci, we can have identical genotypes for the all the loci, what we call identical multilocus genotypes. And this confirmed that what we thought were clonal lineages were identical with microsatellites. So it's a second test indicating that indeed we have some clonality in this species. So this is important because this has consequences of the genetic diversity. I will not discuss it today, but also, sorry, if I go back here, you see that if you don't look at this in your data, you could make wrong inferences. Here, for example, maybe you could think, or you could briefly say, okay, maybe I have diverging lineages, maybe I have different species in my dataset, but not, these are not different species. This is the same species, but with some different clonal lineages, which blur or which bias, as you want, the analysis of genetic structure. Has consequences at the genomic level, but I won't talk about this today. But just to conclude about that, RAD sequencing is interesting to detect clonal lineages. That said, if you are just interested in clonality, and if you already have microsatellites, you can do a very good work with microsatellites for clonality. Another thing is that if you want to study clonality with RAD sequencing, it's important, and I think it's important in any case if you use RAD sequencing, is to include technical replicates. That means it's very important to have, for example, 4 or 5 or 10 individuals that will be sequenced twice. It will give you an idea of the levels of similarity you will get for the same individual. So this is very interesting to understand the mode of reproduction or to identify duplicates. Because sometimes, and I got this with Gorgonians, some Gorgonians who don't do clonality, sometimes you get very similar signal, which means that the same colony was sampled twice. So it can be sometimes underwater. This is not always easy. If you have a dense population of Gorgonians, for example, 2 branches are cut, sometimes from the same colony. So it's important because this will change the analysis of genetic structure. Second example of analysis of population genetics is genetic structure on Mediterranean species. And I will talk in this part about the evolution among depth. For example, if you have the same species, again, my red coral Corallium rubrum, which is present at different depths, we can try to analyze the population size at different depths and also the exchanges among depths, sorry. And if, for example, you have a very important mortality event in shallow condition, this can be useful to study the possibility of recolonization from deep population, what is called the deep refugee hypothesis. So, some words about the red coral, Corallium rubrum. Where is my? I go to this one. Bit exercise. What kind of Anthozoan is it? Easy. Very easy. You have a choice between 2, Exa and octocoral. Already great help. And look at. No? You don't remember what Eric said yesterday? So it's octocoral. You have, maybe it's not easy, but you have 8 tentacles per polyp. This is 1 polyp. And the tentacles have some pinners. Pinners. Okay. So it's octocoral. The red coral. So I think you mentioned that, Daniela, but the term coral can be used for some species in hydrozoans, Stilaster and Lepora, in hexacorals, for example, red coral, and many scleractinians, among others. Another thing you can see here, this is a female colony because we have some larvae. You have male and female colonies. I will go back to that later. So the red coral, here is a map of the known site of red coral. So it's mainly a Mediterranean species with a few populations in the Atlantic. So there are some populations along the Moroccan coast and southern Algarve Portuguese coast. So mainly Mediterranean and mainly Western Mediterranean, a few populations here along the Greek coast, and I guess, yes, if there is a point, there is 1 or 2 cases along Turkish coast. This species, is present from around 10 meters depth. This is what we have around Marseille, which are the shallowest population for this species. The record is around 1,000 meter depth in the Mediterranean, but it's really rare at that depth. It's harvested, so it's the famous red coral which is used to make some jewels. And I would say it's over-harvested for the shallowest population because it's not, if you talk with specialists, they do not say it's threatened by extinction, but in the shallowest population, you have very small colonies left. And this species is one of the species which is impacted by the heat waves, so by climate change, and we had mortality events for the shallowest population in the Mediterranean. Of course, not for below 50 or 60 meters, we don't have impact, but above, we have impact of climate change. So I will not talk all about what has been done on the genetics of red coral in our lab, in the lab of Federica Costantini and others. But previous results have shown that first, with microsatellite, we observe a very local genetic structure, which means that there are some differences between populations a few tens of meters apart, both horizontal, but also vertical. If you take some red coral at 20, other red corals at 40 meters, the microsatellites suggested that these populations are genetically different. And another very important thing is that some experiments have shown that there are some differences of thermotolerance between depths. Just one example of what we did is we took some red coral from 20 and 40 meters near Marseille in aquarium. There was a control and there was a heat shock, short heat shock, quite strong, but there was no mortality, 14 to 24 degrees. And one way to study the tolerance here was to analyze the expression of HSP gene. HSP is for heat shock protein. So these are proteins which are expressed in various organisms when they are submitted to heat stress. And we see that the 20 meters colony have a higher reaction, more expression of HSP than 40 meters. So we really have differences in response to thermal stress between depths. So we wanted to analyze this with word sequencing to better understand the genetic structure among depths. Is it real? And also to test if there are some loci which could be indicative of adaptation to different depths. So I will present just part of the data, but for example, in Marseille, Marseille is here. Sorry, my lab is somewhere here, never mind. And we had 2 replicates to analyze 2 depths in the north of Marseille, 12 and 40 meters, and in the south of Marseille, 8 and 40 meters. And the RADSEC data, it was a PhD of Marine Pralon, had a bit more than 27,000 SNPs analyzed. So, Marine made a PCA, principal component analysis. Each dot corresponds to 1 colony of red coral, and the color indicates the origin, the sampling site. So we have two axes here, the first and second one. The first is the one who indicates who has more information. So the first, the main distinction, if we have this sampling site, is the north of separation. That means that the main distinction, the main separation here is the geographical separation, north-south. And it's not a depth separation. That means that we don't have in the red coral, for example, a shallow population which would be present everywhere and the deep population present everywhere. We have different local population with differences among them, because the second axis, which is maybe not very visible here, but here more visible, is an axis, a depth axis, with a separation among them. So, okay, we can confirm with almost genomic level that we have a genetic structure, vertical genetic structure in this species. And one thing we could ask is if some of these markers, genetic markers, will indicate that there are some differences among them, correspond to support of local adaptation. I will go back to that later. I won't present this, but here, for local adaptation, what is this? So I was quite fast about this, but I will try to explain. Here I have one species, the red coral, the same species, no problem. All our data indicate that 10, 40, 50 meters, it's still corallium movement, the same species. The environment is very different. I can show some data later if you want, but 10 meters in summer, the temperature is higher and more variable than at 40 meters. So the thermal stress probably is higher at 10 meters at 40 meters. So we could say, and we see, sorry, I've shown you that colonies from 10 or 20 meters are more thermotolerant than colonies at 40 meters. So maybe there are some genetic adaptations. Maybe there are some loci, some part of the genomes, which are responsible for the better, higher thermotolerance. So to do that, we can use the rad sequencing data and try to see if the variations for some of these 27 genetic markers, maybe some variations are really correlated with depth or with temperature. So this is an easy way to talk about what we call the search for selected or outlier loci. And there are various methods. I just present two of them here, never mind the names. But with this one, for example, among the 27,000 genetic markers, 82 were candidates, possibly responsible for the adaptation to temperature. Another method detected more than 500 loci potentially linked to adaptation. Okay, and we had another one with other loci. So it's interesting, and it indicates that we possibly have some loci implicated in the adaptation to the different environments, but, and I won't go further on that now, that's things we still want to work on with Sarah also, but there are possibly a lot of false positives. So it's easy with genetic data and with rad sequencing data to use these methods to have some results, to have a nice list of loci under selection, but for various reasons, especially in this species, you can have a lot of false positives. So I present you this result, but I still leave a question marks here because we think that we have to be very cautious about it. Okay. And another thing we have seen is still a PCA here on the same data set, but here is the first axis and here is the, I lost it, fifth axis, I guess. And we did it on the whole data set of marine, including populations from not only Marseille, there was also Corsica and Banyuls in France. And when she did that, she has seen that for all sampling sites, the dataset was separated in almost 2 equal parts. So the first time it was a bit strange. Fortunately, part of the samples, which are the colors here, had been analyzed or had been sampled at the time of reproduction. Maybe it was a sampling in May, June, so the reproduction is June, July. But May, June, it's interesting. Why? Because at that time, the gonads are big enough so that when you open the polyps, you can see the gonads and you can see, okay, this is male, this is female. If you do that in winter, for example, the gonads are too small. Could be done maybe with microscopy, electronic microscopy, but here, when the gonads are very big, just with a binocular, I guess. So for a subset of colonies, we have the sex. These are males, these are females. And so what we identify is that the 2 groups of individuals here correspond to the 2 sexes. And remember that these data are based just on rad sequencing. That means that you have some signal in the genetic data which separates the male and female. That means first that the sex is genetically determined for the species. And second, if we go further, there is a mistake. If we go further in the analysis, we see that as for us, it's a XX, not SY, it's XY system. So the females are X's, the males are XY. Just as us, but it's an independent evolution of this kind of sex-determinant system. So this is interesting. We were able to check that sex ratio was most equilibrated in most populations. And this is useful because now we have molecular sexing protocols, which have been recently used by other labs to sex their colonies when they have colonies which have been sampled outside the production season. We can, with just a bit of DNA, try to determine if your colony is male or female. And finally, I talked about estimating the exchanges among depth. We have not done this yet in red coral. That's what we want to have with Sarah, but we have done this on another species, the red gorgonian. Remember the gorgonian, which was dead, 90% dead, above 30 meters at the beginning. So near Marseille, it's in the Bay of Marseille, we have a very nice population of giant colonies. You see one colony of Parameris clavata. If I'm not wrong, this is Stéphane Sartoretto, which is taller than me, but we have a colony is 1.70, 1.80 meters. You see that's very big colonies. I just want to say that Stéphane did not break the colony. In fact, it was broken by a boat anchor. There are a lot of fishermen who go there to fish as seagull. So we have some threats by anchors. These colonies, these giant colonies, are quite rare, and here these ones are really around 60 meters depth. But nearby, we also have colonies which are much shallower, smaller as well, 30 meters depth. So in this article, which is I accepted also 2 days ago, we add different things: ecological characterization, a bit of experiment to study the adaptation to depth, and also rad sequencing to try to do some demographic inferences. And just to say that if we do this, we can try to estimate the level of gene flow. This is the proportion of genes going in one direction or in another direction at each generation. Don't pay attention to the numbers themselves, but just to say that the information is that the gene flow is very low. So the exchanges among them are quite rare each generation. And there are these are asymmetric. That means that we have more larvae probably coming from 60 to 30 meters, but in the reverse direction. And this would be useful to complement with other populations in the area, but it will be another work. So, last slide, what we are going to do on this population genetic stuff on Gorgonians, there are two things which are ongoing. First, for the red coral, we have a project about sequencing, with red sequencing, analyzing populations along the whole, almost the whole range of these species. So we have samples from Spain, Italy, Greece, which is new. We've got Atlantic and so on, to study the genetic structure, and it's part of FAO project, which is focused to the study of harvesting of these species. So there is genetics, but not only, also some surveys about the levels of harvesting, official levels of harvesting of these species. And at the genome levels, we are going further to study the targets of selection. So we want to produce genomes on these species, and to study hybridization and heterogation in unicellular species. So it's a new project which has been funded this year, where we will do some target enrichment and whole genome sequencing with Sarah, with Alex Baumel in Marseille, with Jean-Baptiste Ledoux in Porto, and with other people. Maybe I forget some of them. So that said, to show you that we go further in the genomic study of this species. And that's it, and a bit of faces of people who work on this subject, and thank you for your attention. If you have some questions about all this. So, oh, I'm late, sorry. So if you are hungry, you'll start. I can try to stop the recording, but maybe it's easier if you... non, je n'ai pas les éteintes, je ne vais pas les rapprocher. Merci.